Hey y'all, this is the video lecture that is for Chapter 13 in the MedSurg book, Lewis's 12th edition. All right, so the topic of this video lecture is genetics and genomics. So when we're talking about genetics, we are talking about the DNA map, the genetic map of humans. Well, actually, any organism has some kind of genetics. Genes are the basic units. The genome is the complete set of DNA that contains all the information for the organism. When we talk about genomics, we're talking about study of a person's genes. And most of the time when we're talking about genomics, we're looking at how genes interact with each other and how they interact with environment. All right, so when we're talking about genetics, we are talking about inheritance specifically. Lots and lots of diseases have at least some kind of genetic component. And we believe that some of the genetics are what makes a difference in how people respond to managing risk factors. So we may have two people who eat the same thing and do the same exercise, but one has heart disease and the other one doesn't get heart disease. You know, there have been histories of people who were long distance runners who dropped dead of cardiac disease. So, you know, genetics plays a huge component in situations like this. Right, we are finding a lot of research into the genetics of disease and genetics of disease development. And most of the nursing organizations have some kind of statement, a policy statement, and expect a certain level of education and certain understanding of genetics for professional practice. All right, so just to review from basic high school biology, humans have 23 pairs of chromosomes. One of those pairs is the sex chromosome. So the 22 pairs are the same in males and females. The X and Y are the sex chromosomes. That's what makes us biological, male or female. Female has two X's, male has X and a Y. And we inherit half of our chromosomes from each parent. There are about 30,000 genes in a person's genome, and they control every interaction, every function within every cell. We know that we actually share an enormous amount of genetic material, an enormous amount of DNA is exactly the same in the other great apes. In fact, chimpanzees, we share over 98%. Gorillas, we share over 98%. And bonobos, who are the chimpanzees' closest relative, we share more than 98%, almost 99% of our genetic material we share with the great apes. The difference is what's turned on and what's turned off. Because sometimes, we have lots of genes that are put on hold. Proteins, the genetic material controls protein generation. So the genes are actually kind of lined up along a chromosome, and that chromosome is twisted and then twisted again, the way it's stored. Otherwise, it would be enormously long. We call it an allele, those pairs of genes that happen on these chromosomes. They may be called alleles. We have dominant and recessive. So dominant, if you got the gene, you got the thing. Recessive, you have to have more than one copy of something, and if you've got the dominant allele, you may not see it. So for example, my mother had green eyes. My dad has brown eyes. I have brown eyes because I got green from mom and brown from dad, and brown plus green equals brown. My daughter has green eyes. She got green from her dad, and she got a green from me because I can think about myself as being a carrier for green eyes. So she has the most beautiful olive-colored eyes I've ever seen. So that's an example of a recessive versus a dominant. You'll also see the terms genotype and phenotype. So genotype is the list of the actual genetic makeup of a person. It's their genome. Phenotype is those physical traits that we see. So my genotype drives a phenotype. So the fact that I have my genotype is whatever the genetic thing is for brown eyes and green eyes, but the phenotype that you see when you look at me is brown eyes, because that's the physical trait that you can see, that gets expressed. So here's a lovely little diagram of chromosomes. They live in the nucleus of the cell. They are protected by the nucleus from everything else in the cell. The DNA on the left, you can see it comes in pairs. And then it's twisted, but then that whole molecule is twisted around kind of like a telephone cord. Makes it much shorter. So DNA stands for deribonucleic acid. There are sugars on the outside, and then there are 4 bases, adenine, guanine, cytosine, and thymine, that store the code, and it's the order of those that come together that makes the information. The other thing that you should kind of keep in mind is that every cell has the exact same DNA. They're expressed differently in the different kinds of cells that the cells grow up to be, but they contain the entire genetic reference points for those for the person. Different bases pair up in different ways. So A always pairs with T, C always pairs with G. And then we have ribonucleic acid, and ribonucleic acid is the one that actually that copies DNA and takes that map, that template for whatever the protein is, out to the cell so that the cell can make it. It only has a single strand, where DNA has a dual strand. When the body needs to make a protein, a cell needs to make a protein, it untwists the DNA. The bases unlock, and RNA comes in and makes a copy, or makes the other half of the copy. Then it can take it out to the cell, and the cell will reconstruct that protein. That process is called transcription, and when they're arranged when it's complete and it's that DNA, whatever protein needs to be made based on the DNA template, is released into the cell and it's able to function, then it's called translation. So transcription, you can think about copy, and translation is putting it into use. Just like when you translate a language, you're putting words into a new use. All right, so once again, back all the way to basic biology. We have different principles of cell division. So mitosis, when a cell divides mitotically, it turns into, they are genetically identical. They only divide once, and then they're exact replicas of each other. Some of our germlines, particularly the X chromosome and Y chromosome, the eggs and sperm, will divide twice in rapid succession. So sex cells only have, instead of having 2 copies, 2 copies of every chromosome, they only have 1 copy, because they're going to get 1 copy from the other parent cell, the other reproductive cell. So you would get half from each parent, and then you would have a complete set of chromosomes that can then divide, become a trophoblast, embryo, and then a fetus. All right, so when we talk about mutations, we are talking about any change in the DNA sequence. It can be a single change, it can be multiple changes. They can be inherited from a parent or they can be acquired. So I may have some genetic mutations that happen during cell division on my end, or I may have a mutation that I inherited from a parent. All right, so different types of mutation, like I said, inherited from a parent, we call that a germline mutation. Goes through one of the reproductive cells and it occurs in every cell in the body. Somatic mutations are those that are acquired. They don't get passed on to the next generation. Okay, they happen in a particular group of cells. So perhaps someone has been exposed to radiation and there are several cells that have a mutation. Any cell that results from that cell, that as mitosis occurs, it will get copied through. If it doesn't get, if that mutation does not involve an X or a Y, it will not get passed on to the next generation. So we talk about inheritance patterns as either autosomal dominant, where you only need one pair of that gene to have the phenotype, or autosomal recessive, where you, to be able to express the disease, you have to have two gene pairs that match. This is where we say that someone is a carrier. They have a gene for it, but they don't have enough genes in order to have the disease or have the phenotype. Now, if they are reproducing with someone who is also a carrier, then children from that union may actually have enough of the gene pairs to have the phenotype of the disease. Then we have some X-linked recessive disorders. So X-linked are things that happen on the X chromosome. Most of the time our X-linked recessive disorders, these happen to boys and men because they only get one shot at an X chromosome. That whole X chromosome is, they only get one shot at it. Women tend to not have X-linked recessive disorders. They would have to have two chromosomes with the same recessive gene on it in order to experience the disorder itself. They can give it to their kids if they have a male child, because once again, they're only getting that, they may only get an X with that defective chromosome on it. Y-linked disorders, those are transmitted through the Y chromosome. Those are only ever affecting males. So here's just some examples. If you look on this family pedigree, this is a kind of a standard way of looking at these things. So on the legend, you'll see that a square or rectangle is male, the oval is female. They're filled in if they have the disorder, and they are half filled in if they are the carrier because they've got one gene but not enough to have the disease. So the autosomal dominant, you can see on the on the far left. The male has the disorder, the female does not, and of an autosomal dominant disorder or autosomal dominant genetic trait, 50% of those offspring are going to have or could have whatever is that characteristic. With two carriers, 1 in 4, a child has a 1 in 4 chance, a 25% chance of having the disorder, 50-50 on getting being a carrier, and then 25% chance of neither being a carrier nor having the disorder. Okay. And then there's the final example is that X-linked recessive. So mom would be a carrier, dad would be disease-free. In this situation, The male children may be, they'll either have it or they won't, one or the other. They're 50-50 as whether or not they'll get that. Then female children is 50-50 whether or not they'll be a carrier. And we can expand these inheritance patterns to multiple generations. That's the point of this one, is they've added yet another generation to the potential for disorder. All right, genomes were mapped back in 2003, and everybody thought this was such a big deal, and we were going to be able to do everything very quickly. But the scientists said then, and continue to say, that even though we've learned a lot, there's so much more that we don't know. There are opportunities for continuing to learn about genetic predispositions. One of the things that we have been able to do is to match organ donors with recipients a little bit better, because we can get their genetics closer. The closer we can get the genetics, the less likely a patient will be to have a transplant rejection. All right, we talk about mutations, and some mutations, some disorders are pretty straightforward. Those autosomal dominant, where if you get the mutation in a single gene, you get the thing. People are usually not that straightforward. So a lot of times, some of these genetic or family linked kind of things that we do, there may be mutations on in multiple genetic components. So it's really hard to nail down one single gene that is responsible for something. All right, so some of our single gene disorders, either autosomal dominant, recessive, or X-linked, they're pretty rare, but they do happen. Some are more common than others. So cystic fibrosis is one. So I'm going to go through these a little bit differently than your slide does. Looking at autosomal dominant characteristics. I'm going to say characteristics because, thinking about other things as well. Not everything that is inherited is a disorder. Things that are diseases that are autosomal dominant. So meaning if you get one copy of the gene, you get the thing. Marfan syndrome, Huntington's disease, which we'll talk about in care 2. Achondroplasia, which is one of the most common forms of dwarfism, is autosomal dominant. And there's a disorder called osteogenesis imperfecta that would get kids with enormously fragile bones. Those are autosomal dominant. And like I said before, brown eyes are autosomal dominant. When we're looking at autosomal recessive, things that would be autosomal recessive with a single gene kind of pattern. Cystic fibrosis is kind of that way. You got to have 2 versions, but it's a very small, it's only 2. Sickle cell disease. You can have sickle cell trait and be a carrier. Full-blown sickle cell disease, though, is autosomal recessive. And red hair is actually autosomal recessive. Then X-linked, things that are X-linked. Red-green color blindness is X-linked. So typically we think about men being colorblind, and red-green color blindness. Occasionally you will have a female who gets an X, who has a mother who is a carrier and her dad shares the X with her that also has it. So I have a friend who was a med-surg nurse, and she could not tell the difference between red and green. Duchenne muscular dystrophy is also X-linked. And hemophilia A, which was running rampant through Queen Victoria's offspring and grandchildren, Hemophilia A is an autosomal recessive X-linked. All right, there are some that are, we tend to see inherited conditions that involve more than one disorder. These are complex things. We also have environmental factors that add into risk for developing these diseases. And then we have some rare hereditary mutations. So the breast cancers, the BRCA1 and BRCA2 genes. Right, when we talk, if you hear the term epigenetics, epi has to do with things going around that surround something. So an epidemic is stuff going around the world, right? It's a disease going around the world. Epigenetics look at inheritable changes in how genes are expressed. They don't involve the DNA sequence itself. So what we're looking at is what is the difference in what we see, not in the genotype itself. So chromosomal disorders, there are a lot of them, and we will talk about some of these when you guys get to maternal newborn, because we do talk, this is usually where we kind of see these things. Like Down syndrome, that is an extra copy of a chromosome. So instead of having a pair of chromosomes at chromosome 21, they actually have 3 versions of that chromosome. So that's one of the trisomies. There are some other trisomies. Trisomy 13, that's very rare. Those kids usually don't survive past a few years. Then we also have some, we call them translocations. There is a chronic version of leukemia where there's just a flip in where some chromosomes are. They're all there, they're just in the wrong place. Okay, so we have genetic screening, and we do this, we don't do genetic screening on every person on the planet, on every person in practice. But we have genetic testing. So there are some screenings that we do. We do screen pregnant women for, and it's a very, it's very much a very screening. It's not diagnostic. It's just giving us an idea whether or not we should continue to dig around and look. One of those is alpha-fetoprotein. That's part of a selection of labs that we do on a first prenatal visit. We'll talk about those in maternal at newborn. But if there's a specific reason to test someone, if we have a screening test that comes back, if we have a patient who has the BRCA gene, it may be advisable to at least offer testing to their offspring. So a daughter of a woman with breast cancer that we know has the BRCA1 or BRCA2. There are several different kinds of genetic tests that we can do this. We can test for sickle cell disease or sickle cell trait. We can test for these things, and then patients and their families can make their own decisions about how they want to proceed with that. I've had patients who found out that they were positive for the breast cancer genes and elected to have bilateral mastectomy. If it's not there, it can't give me cancer. So there are pluses and minuses. Some people do not want to know. They just don't want to know. They don't want to change their lives in a good way or a bad way. They don't want to spend their lives looking over their shoulder trying to decide if that ache, pain, lump, bump, whatever it was, is this disease coming out. So there are pluses and minuses to submitting to genetic testing. It is useful in healthcare. It does help us tailor medications to different patients. things that people worry about is insurance canceling because you've got some gene. Actually, that is against the law, and there is a federal law that prohibits discrimination against genetic information. And genetic information is actually covered by HIPAA. So there are some protections that are set into place. But things, family effects, I've talked about these already. There are implications for those who are not tested. Sometimes you find kin folks that you didn't expect to find that can be disruptive to family relationships. Then making sure that we're interpreting and helping patients interpret some of these tests positively and in a positive way. All right, so if they're positive, they say they're positive, they have the disease, they have the gene. If it's negative, they're not affected by a disorder and not a carrier. Oh, and a lot of insurance companies do not cover the cost of genetic screening. There are some direct-to-consumer genetic tests. So Ancestry.com and 23andMe are kind of the most common ones that we see. Some people just do the ancestry component. Some people do the full health risk component as well. Patients may bring these to the office and say, I don't understand. This says I'm going to get whatever. And it's important that we help them understand what some of these things mean. Right, we can do DNA fingerprinting because genetically everyone is different. The only people who have identical DNA are identical siblings, so identical twins, triplets, whatever the relationship is, but they must be identical. Fraternal twins are no more similar genetically than two separate kids. The way I explained it in OB is they are just like two separate kids. They just shared a bunk bed until they got here. They are no more genetically linked than any other sibling pair. We can get DNA from any cell. We can get it from hair cells that are, for the root of the hair. We can get it from saliva. We can get skin swabs, cheek swabs, blood, anything that we can use, anything that has that patient's cell, we can use. We do use these, and I think we hear about it most often with forensics. So patients that were incarcerated years ago, and now we can do DNA testing and say, yes, that evidence did not belong to this person, and we can, free someone who has been wrongly accused, wrongly incarcerated. Paternity testing is based on DNA, and some of our HIV testing also involves DNA fingerprinting as well. Pharmacogenomics and pharmacogenetics. So you can see they've put pharmacology and genes here together. And so pharmacogenomics looks at how drugs respond based on the genetics of the patient. When we're talking, it's broader than pharmacogenetics, which looks at drug responses in response to a single gene. So there are drugs that are metabolized in the liver, and depending on this whole set of enzymes called cytochrome P450, there's a whole bunch of subcategories of that. But cytochrome P450 breaks down a lot of our drugs. And some patients with a particular level of cytochrome P450 may have increased or decreased response to different medications. So here's just kind of an example. Different genetic variants. If you have Some people may actually need a higher dose of something because of their genetic variant. Some people may have a drug at a lower dose because they don't metabolize it as well. Warfarin is one of those that is very sensitive to cytochrome P450. Another one is codeine. And one of the things we know pharmacogenetically, is that 10% of the Caucasian population cannot metabolize codeine into morphine because of cytochrome P450 variants. So when they say codeine does nothing for me, codeine works because the body can turn it into morphine. 10% of the white population can't do that. So very, very highly likely that they are telling you the truth. One of the things that we've been looking towards is, can we fix a disease by fixing the genetics of that patient? There are a couple of disease processes that are approved by the FDA, but for the most part, this is still highly investigational. Stem cell therapy is also another therapy based on genetics. Stem cells are kind of root cells. They're the cells that everything comes from. They can have different levels of what they can grow into. So totipotent, they can become anything. You think total, down to unipotent. They've developed to the point where they can only become one thing now. So why is this important? We want to make sure that we can assist in providing information to patients, because some of our patients are going to need to make decisions based on genetics, like I was talking about earlier. Patients who get testing and they find out they've got a really high rate, high likelihood that they will develop a kind of cancer. Patients who find out that they are carriers for a disease may want to make decisions about reproduction. Patients who are expecting a child that they know has a genetic disorder may want the option to make a choice about whether or not to terminate that pregnancy or to carry that pregnancy to term. So there are a lot of places where where genetics can help people figure out what it is they want to do. But it also means they have to make some decisions and confront some information. There are certifications in genetics. You can be an advanced practice nurse with a specialty in genetics. You can be just like you can be a certified med-surg nurse, you can be a genetic clinical nurse. Right, so when you're doing a family history and you're looking specifically at genetics, you can make your own pedigree. And usually we look at 3 generations, so we go 3 generations back. And then there are agencies like CDC, they have a way to do that. And the person that you're interviewing, you start with that person, and that person is called the proband. And so things that we're going to look at when we're constructing the pedigree, we want to look at any medical illnesses they've got, cause and age. If we're looking at someone up or down in the list, are they, what age did they die? What did they die from? Things that we would look at, we look for things that affect or don't affect genders. We look for disease in more than one close relative. So if we're looking at a patient with breast cancer, and we're seeing a lot of breast cancer in that family, we kind of can map some of that out. Right, so I think everybody has kind of done this kind of thing in biology way back. So this is autosomal recessive, autosomal X-linked recessive and autosomal dominant. It really does kind of put a different graphical design on what we were looking at earlier with the pedigree tree. If by doing that pedigree, we can actually see some patterns of inheritance. We want to make sure that those patients get referred for further screening should they want it. We want to make sure that we are treating that patient with respect and being supportive, making sure they understand what their decisions are, what decisions they can make, and just being supportive as they get information. All right, so let's look at this one. Which patient would the nurse identify as most at risk for developing a genetic-related disorder? Okay, so let's go through these one by one. So if we look at A, a 26-year-old woman who has a history of 3 miscarriages, 3 spontaneous pregnancy losses. There is nothing about that that will make her have a genetic-related disease later on. Okay. Let's go down to C. An 18-year-old man who is a carrier for an autosomal recessive genetic disorder. Autosomal recessives do not express into the phenotype. So he is a carrier. He can pass it on to his kids. However, he is not going to get sick. He is not going to develop a genetic-related disorder from being an autosomal recessive carrier. 32-year-old woman who reports her mother died from a serious drug reaction. I got nothing in there that says genetic-related disorder. However, let's go up to B. A 40-year-old man who reports his father and 2 sisters have type 2 diabetes. We know that there are some genetic links with diabetes. He already has diabetes. He already has multiple people with diabetes in his family, including a parent and 2 siblings. So out of all of these 4 patients, that is the one who has the highest risk of developing a genetic-related disorder. All right, let's look at one more. We have a male and a female who are both carriers for sickle cell disease and express concern about having children with the disorder. So which response would be most appropriate? Okay, so let's think about let's think about sickle cell. And we know that sickle cell is autosomal recessive. So someone would have to inherit from both parents. So if mom and dad are both carriers, then there's about a 25% chance that a child from those 2 people will have the disorder. So it's D. Option A is just a terrible thing to say because you're telling them, oh, you don't need to have kids. And that's not necessarily, first of all, it's not our choice. Second, we're making a decision for them and they need information. They don't need to be pointed somewhere yet. One parent, no, you get the disorder if you're a kid from a carrier, for both carriers. And we can't say that they'll most likely be born with the disorder. So the way it works is 25% chance they won't inherit the carrier status, 50% they'll inherit a carrier status, 25% that they would have sickle cell disease. All right, last question. So we have a nurse caring for a patient who is prescribed clopidogrel or Plavix. The nurse is most concerned if the patient states, and we want to take a look at these. Okay, so let's talk through all four of these options. So A, I was diagnosed with an inherited genetic disorder. That could be anything. That could be anything. So that doesn't concern me as much as some of the other statements concern me. I had a severe allergic reaction to warfarin. We don't inherit specific allergies. We inherit the tendency for allergies, but we don't actually inherit antibodies. So I'm going to put a pin in that one. Plus, warfarin and Plavix are two completely different drug categories. Very unlikely that there is even any overlap. C, my tumor gene was negative for the production of HER2. Okay, doesn't really have anything to do with clopidogrel. However, a genetic test showed a low level of an enzyme in my liver. Right, cytochrome P450 is a liver enzyme. So of these four, I would be most concerned. I would want to explore exactly what that was, what that patient was talking about, as far as pharmacogenetics of that patient. Okay, guys, that is all for this lecture. If you have any questions, send me an email, catch me in the hall, and we'll see if we can get it sorted out. Thanks.